Welcome to G Fun Facts Online. Today, we're delving into a topic that affects all of us, a hidden world of viruses and the unseen bridges they cross to enter our lives. We're talking about zoonotic spillover, how pathogens leap from animals to humans. This isn't a rare fluke of nature. In fact, over 60% of all known human pathogens originated in other animals. Major diseases, from Ebola to HIV, began this way. But how does it happen? And what happens when a virus that's perfectly adapted to one animal suddenly finds itself inside a human? Today, we'll explore the mechanics of spillover. We'll meet the animal kingdom's most surprising viral superstar, and we'll follow the trail of one of the deadliest emerging pathogens on the planet, a virus that serves as a chilling case study for our times, Nipah virus. The story starts in what scientists call a reservoir host. This is an animal species that naturally carries a pathogen, often a virus, without ever getting sick. They are a permanent, circulating source of the virus. The spillover happens when we, or sometimes an intermediate animal, come into contact with that reservoir. This contact can be direct, like touching an infected animal or its bodily fluids. It can be indirect, like eating food or drinking water contaminated by that animal. It can be vector-borne, where a tick or mosquito bites an infected animal and then bites you. Or it can even be through inhaling tiny airborne droplets. But for a virus to make the jump successfully, it needs more than just an opportunity. It needs a key to our cells. The virus has to be able to bind to receptors on the surface of human cells to get inside and replicate. Many of these spillovers are dead ends. The virus infects one person but can't spread any further. But sometimes, a virus adapts. It gets better at replicating in us. It learns how to spread from person to person. And that is when you have the makings of an outbreak, or even a pandemic. Now, when you think of animal reservoirs for disease, you might think of rodents or birds. But one of the most significant, hosting a disproportionately high number of dangerous zoonotic viruses, is a creature of the night, the bat. Bats are the reservoir for the coronaviruses behind SARS and MERS, as well as Ebola, Marburg, and the deadly virus we're focusing on today, Nipah. So what makes bats the perfect viral reservoir? It's a combination of their unique biology. First, they have a truly remarkable immune system. It's in a constant state of high alert, always primed to fight viruses. This allows them to suppress viral replication so they don't get sick. But it also creates an evolutionary boot camp for the viruses they carry. It forces the viruses to become stealthy and persistent to survive, making them more robust. Second is the power of flight. The intense metabolic effort of flying raises a bat's body temperature to levels that would be a high fever in any other mammal. They essentially experience a daily fever, which helps them control viral infections that would be lethal to other species. Third, their social behavior. Bats often live in massive, densely packed colonies, sometimes numbering in the millions. This is the perfect environment for viruses to spread rapidly and be maintained within the population. Finally, they are long-lived for their size and can travel huge distances, effectively carrying and distributing their viral passengers over vast geographical areas. This combination of factors makes bats a perfect storm, a menagerie of viruses well adapted for survival and ready to spill over into any species like us that lacks their specialized defenses. And that brings us to the Nipah virus. The story of Nipah is a stark lesson in how human activities can shatter the natural boundaries that separate us from the animal kingdom's viral library. In late 1998, in the state of Perak, Malaysia, a mysterious illness emerged among pig farmers. They suffered from a severe fever that quickly progressed to encephalitis, or inflammation of the brain, leading to confusion, seizures, and a high death rate. At first, authorities suspected Japanese encephalitis, a common mosquito-borne virus. But the clues didn't fit. The disease was also ravaging the local pig population, causing a highly contagious illness nicknamed barking pig syndrome. And it was spreading mainly among adults who had direct contact with sick pigs, not through mosquito bites. An intensive investigation led to the isolation of a brand new pathogen, a previously unknown virus from the same family as the Hendra virus in Australia. It was named Nipah, after the village where the first sample was taken, Sungai Nipah. So what happened? The puzzle was pieced together, and it was a textbook case of spillover. The natural reservoir was, you guessed it, local fruit bats. But the virus didn't jump directly from bat to human. It used an intermediary, an amplifier host, pigs. The critical driver was the expansion of industrial pig farming into areas bordering the bat's natural forest habitat. Mango and other fruit trees planted near these massive pig farms attracted the fruit bats. As the bats feasted at night, their saliva and urine, laden with Nipah virus, rained down, contaminating the pigsties below. The pigs, crowded together in high-density pens, were the perfect amplifier. The virus spread through them like wildfire, and the huge viral loads in their respiratory secretions created a bridge for the pathogen to jump to the farmers. The outbreak led to over 280 human cases and more than 100 deaths. It was only stopped by the heartbreaking but necessary culling of over 1 million pigs, causing devastating economic loss. Just a few years later, Nipah re-emerged, but in a different place and with a new, more frightening pattern. Starting in 2001, annual outbreaks began in Bangladesh and neighboring India. These outbreaks were even deadlier, with fatality rates often exceeding 75%. And crucially, there were no pigs involved. This time, the route was more direct. The culprit was a popular local delicacy, raw date palm sap. Fruit bats also loved this sweet liquid. They would climb the trees where collection pots were hung and lick the sap, contaminating it with their virus-laden saliva. Unsuspecting villagers would drink the fresh, unpasteurized sap and become infected. This seasonal pattern, tied to the winter sap harvest, became a hallmark of the outbreaks in this region. But here is the most alarming part. The strain of Nipah virus in Bangladesh and India appeared to be better at spreading between people. While human-to-human transmission was limited in Malaysia, these new outbreaks saw significant spread, especially among family members and healthcare workers caring for the sick. This occurs through close contact with a patient's bodily fluids, like respiratory droplets. This heightened transmissibility makes this strain a grave public health concern, bringing it one step closer to what's needed for a large-scale epidemic. An infection with Nipah is a terrifying ordeal. After an incubation period of about 1 to 2 weeks, the initial symptoms are nonspecific: fever, headache, muscle pain, vomiting. It looks like the flu, but this can be followed by severe respiratory problems and, most characteristically, a devastating neurological attack. The virus triggers acute encephalitis, leading to dizziness, drowsiness, and a rapid descent into a coma, sometimes within 24 to 48 hours. The case fatality rate is estimated between 40 and 75%, and in some outbreaks, it has approached 100%. For survivors, the battle isn't over. Many are left with persistent neurological problems, like seizures. And disturbingly, some patients can experience a fatal relapse months or even years after they've supposedly recovered. To this day, there are no licensed vaccines or specific drugs to treat Nipah. Care is limited to intensive supportive care. This combination of high mortality, human-to-human spread, and no specific treatments is why the World Health Organization has classified Nipah as a priority pathogen with pandemic potential. The emergence of viruses like Nipah isn't just bad luck. It's a predictable consequence of a world thrown out of balance by human activity. The single greatest driver is the destruction of natural habitats. When we clear forests for agriculture, logging, or urban expansion, we force wildlife into smaller areas, pushing them into closer contact with us and our livestock. This stress can also compromise their immune systems, causing them to shed more virus. Furthermore, a biodiverse ecosystem has what's called a dilution effect, where a virus gets diluted among many species that aren't good hosts. When we destroy that biodiversity, the species that thrive are often the ones, like certain bats and rodents, that are excellent reservoirs for human pathogens. The Malaysian Nipah outbreak is the textbook example of this process in action. The second major driver is agricultural intensification. Modern factory farms with thousands of genetically similar animals crowded together are the perfect environment to amplify a virus. When a virus spills from a bat into a pig, that pig farm becomes a viral factory, producing enough virus to easily infect humans. The third driver is climate change. As the world warms, animals and the insects that carry disease, like mosquitoes and ticks, are shifting their geographic ranges. They're introducing pathogens to new places where human and animal populations have no immunity. The reemergence of Nipah, SARS, Ebola, and COVID-19 are not isolated incidents. They are symptoms of a deeper problem. Human-driven environmental changes are dismantling the natural barriers that once kept these dangerous pathogens at a safe distance. So, how do we confront this threat? The old way of waiting for an outbreak and then scrambling to respond is a proven failure. We need to shift to a proactive, integrated approach. And the core of this strategy is a concept called One Health. One Health recognizes that the health of people, animals, and the environment are all interconnected. It means veterinarians, ecologists, public health officials, and policymakers all working together. For Nipah, that means tracking bat populations, ensuring biosecurity on farms, and educating communities about risks like contaminated date palm sap, all at the same time. The second pillar is building a global immune system through surveillance. We can't fight what we can't see. This means actively looking for new viruses in wildlife and livestock, not just waiting for them to appear in people. It also means using cutting-edge genomic surveillance to rapidly sequence a virus genetic code, allowing us to track its spread in real time and spot dangerous new mutations. The most effective approach, however, is to prevent the fire from starting in the first place. This means tackling the root causes. Protecting and restoring forests is a public health imperative. It rebuilds the natural barriers that contain viruses. We need to transform agricultural practices, improving biosecurity, and reducing the extreme density of animals. And public awareness is key. Simple things, like campaigns in Bangladesh to encourage boiling date palm sap before drinking it, can save lives. Finally, we need a last line of defense. While prevention is the goal, we must be ready to respond. The world is in a race to develop a vaccine for Nipah. Several promising candidates are in development, which could protect frontline healthcare workers and vulnerable communities. We also desperately need effective treatments, like monoclonal antibodies, that could dramatically reduce the disease's terrifying mortality rate. The story of Nipah virus is a chilling reminder of our vulnerability. But it is also a call to action. Zoonotic spillovers are not random acts of nature. They are a reflection of our relationship with the planet. Preventing the next pandemic isn't just a job for scientists. It is a shared responsibility that requires a global commitment to protecting our environment and recognizing that the health of every species, including our own, is linked. The future of our health depends on it. Thank you for joining us on Fun Facts Online.